And our next speaker will be Erica Ollman Saphire from the La Jolla Institute for Immunology. And she will be telling us about her work on a global consortium, next generation SARS-CoV-2 antibody therapeutics and stabilized spike.
Thank you. So I'm a structural biologist and also a virologist and an immunologist. And what I'd like to tell you about today is a structural biology directed global collaboration to understand what the right therapeutic should be and if we had the right research pipeline in the first place to find them. So let me share my slides with you. What I'm going to talk about is an Immunotherapy Consortium, so using antibodies as treatment therapies against the novel coronavirus. And this is a global collaboration funded in 3 parts by the Gates Foundation, the GHR Foundation, and NIH. The goal of this consortium was to do a side-by-side analysis of all of the candidate therapeutics that started emerging forth in the last year. We're looking at well over 350 now. To compare them side by side in standardized ways and in standardized platforms to understand which ones are really best, what features made them best, which in vitro assays best forecast in vivo success, and if we needed a combination therapy, or what we call a cocktail, what's the best combination we could make? Because ultimately what we want to find are treatments that are potent enough to be low dose and affordable throughout the world in low and middle income countries. Now, the idea for this coronavirus consortium that we were asked to run came from work that we had done in Ebola virus. That my lab was founded to work on Ebola virus and other emerging infectious diseases. And we had done a lot of X-ray crystallography, understanding what the surface glycoproteins of Ebola and the related proteins are, how human antibodies from survivors would interact, what the right sites were by which antibodies could inactivate or neutralize the virus, and what parts were conserved and what parts were variable. So we'd done all of this work and we understood what the structures were, and we had antibodies that we knew would neutralize, and we thought we had a good handle on the physical mapping of form to function, of site to activity. But there was something else. Back up to 2013, we realized we were missing something as a whole research field. Now, 2013 was in 1 year before Ebola virus exploded from a single infected toddler in Guinea to a 30,000 person outbreak with 11,000 deaths. At that point in time, 2013, this was the state of the research field. We had antibodies that neutralized beautifully in cell culture, but they did not protect the non-human primates. The monkeys all died. We had other antibodies. I'm showing you here 3 that bind the upper and outer region. Some are modeled by negative stain, one's an X-ray structure. That combination didn't neutralize in vitro. But you put them together and it did protect all the non-human primates. And so on the one hand, we have something that looks beautiful in vitro and fails in vivo, and something else that looks fabulous in vivo, but didn't register as interesting in vitro. So we're clearly missing something. We're missing some information. Either this data that the research, this is the entire research field that had come to this point, either what this data was telling us was that we needed a mixture and not a single therapy, or it was telling us something about the immune signaling because we had some ideas about how different glycans on that FC of the IgG would inspire better, worse protection, or something about the way we were measuring neutralization and cell culture wasn't adequately forecasting what would really protect living things. We want to protect people from Ebola virus, not tissue culture plates. And so we thought as a research field, we needed to understand what it was that led to protection of living things. And we needed a statistically significant pool of samples to do so. The field was small at the time. Maybe we'd come up with the exceptions and not the rules. And so that was the question before us. And this was the idea that we had in lab meeting one day. And the question was, how do we go forward from there? It was clear that we as a body of scientists in the Ebola virus field were going to need better tools, and we needed to look at more samples. We can't draw a broad conclusion from looking at 12 things. We needed to look at hundreds. And the issue was that every lab was working in a silo, using their own techniques and on their own pool of antibodies, and a pandemic had begun. The cases were doubling every 2 weeks, and we needed to do a much better job. What we as scientists needed was a research framework that would let us do that better job, a research framework that would let us do the sample sharing that we knew we needed and the data sharing that we knew that we needed within an environment that is fundamentally built on competition. The research enterprise is built on competition. Competition drives us to get results faster. Competition drives innovation. Competition inspires you to do a better job and up your game. There's also intellectual property. We know that we need to share samples and information, but sometimes even if the scientists had wanted to, their university wouldn't let them because they need to protect that IP. And so how do you do what you know you need to do, which is involve more people, more ideas into a broader study within a framework that tends to promote single efforts instead? How do you enable collaboration in an environment built in competition? And we put a lot of thought into this over the years. And what we did is we built a study that, first of all, blinds the samples. So everybody can contribute their molecules and nobody knows whose are whose. They're just code named, big 1, big 28. BIC is the Viral Immunotherapy Consortium. We had ironclad agreements to protect intellectual property, whereby everybody's data and everybody's molecules were preserved. They were free to publish whatever they wanted to publish on them. And what we would be studying as a field was the aggregated data of what do antibodies do against these viruses. And we would run parallel tracks to get around the other issue. The time it takes to organize people into a group can slow you down. In an escalating pandemic, you also need speed. So we would run parallel research tracks, some hurtling forward to race virus and other ones doing the slower, more thorough, comprehensive analysis. That was funded by an NIAID CETR, and it launched a global collaboration. And ultimately what we put together were 44 previously competing labs in 5 continents that come from industry, academic, and government walks of life into a single comprehensive effort to figure out together what antibodies are best and how we would find them better next time. Over 3 years, we together, about 100 people, looked at 200 different monoclonals, each the very best or very favorite from every different lab that would be erasing them in different ways and analyzing them in different ways. And so in the vertical axis, these are the samples we looked at. In the horizontal axis, everything we measured about them by taking the shared pool of blinded antibodies and sending them out to different experts, each of whom would do something different: structural biology, glycan chemistry, different animal models, different neutralization assays, different immune assays. And we could put together big data models, these are from Christian Andersen, the Scripps Research Institute, of what variables correlated with other variables, which kinds of things we could measure high throughput in vitro would track together. And then we could build regression models to understand which of the things that we could measure high throughput, cheaply, rapidly in plastic actually would forecast in vivo protection. And the answer was you need 2 things. You need mechanical inactivation of that molecule of the virus that drives entry. That's neutralization. But we could figure out which ways of measuring better neutralization were better forecast than others. And you need that antibody to recruit the immune system to come in and clear the pathogen from the body. So we needed those 2 things. That work ultimately resulted in 78 peer-reviewed papers, 10 PhD granted, and we could revise the research pipeline by now having together on everyone's samples and with everyone's and everyone's input, figure out which in vitro assays were most effective. And we moved 6 new therapies toward the market where none had been before. In 2020, we were asked to reconstruct the same thing against SARS-CoV-2, but twice as big, twice as fast, and now with a profit motive. Because frankly, no one was ever going to make any money on Ebola virus. Everything needs to be donated. Here we're up against a bigger challenge with major multinational corporations that had intellectual property that we needed to work with and protect. So how are we going to do that? The major question when we began was, would people participate? Because there was suddenly a major global market in America and Asia and Europe, as well as in emerging areas of the world. And could we do this in a timely manner? And so we worked pretty hard, and we now have well over 350 different molecules that have been contributed by dozens and dozens of different labs, discovery engines, and some of them came from survivors of this pandemic. Some came from survivors of SARS-CoV-1 in 2003 that had been re-engineered for this. Some came from immunized animals. Some were designed in silico. So they came from lots of different ways. And we looked at each one of these molecules using multiple different assays, including structural biology, to figure out what we could learn about these antibodies and what it was that best correlated with human clinical success. So our contributors come from industry, academia, and government labs. 2/3 of the molecules are actual candidate therapeutics from companies. Some of the companies are the major multinational corporations and the household names. All the molecules in the NIH clinical trials are in here, as well as multiple small startups, and it gives them a chance to have their molecule examined on the same level playing field as everybody else's. And this is what it looks like to us. This is our lab. All the samples are blinded, and everyone's competing therapeutic is given a code name. So everything has an equal footing, whether it comes from an academic lab or a major company. The samples come in, they're blinded, they're aliquoted, they're sent out to research partners, each of them doing a different kind of analysis on this. And the data is aggregated together in a COVID-DB database housed at La Jolla Institute for Immunology. And the contributors get something back. Their intellectual property is protected. All of the data we get, and we can do a lot more assays and different levels of biocontainment than they have. For example, they may not have the structural biology tools we have. They get all that back, and they can use that for their own investigation on the drug filing. In this database, we now have immediate publicly available information on the hundreds of different therapeutic candidates, and these can be downloaded and put into Excel spreadsheet for different researchers to use for their own purposes. And so that was a study that we were building and propelling and working on the agreements for when the variants began to emerge. We were asked to pivot that. And so that's the structural biology that I want to show you today. The first thing they did to make sense of these 350 different competing antibodies was a competition analysis. We thought in order to outcompete viral escape, we were going to need molecules that hit at least 2 different footprints so that any one point mutation wouldn't knock out the pair, or that the 2 together would exert enough pressure to limit mutation. If we need a cocktail, we need an A and we need a B, we need an A and a B, they're going to play well together, not compete with each other. And so Cartera did for us a high throughput surface plasmon resonance mapping of the antibodies in the consortium, and they could separate them by competition into major groups. Those against the receptor binding domain, where one gets highly potent neutralization, can be divided into 7 major communities. Those 7 communities kind of look like this in a grid of how much they compete or don't compete with each other. The first major division of competition separates what we call groups 4 and 5 from the others. And ultimately, the level of competition, as you might expect, correlates with the geographical space. Groups 1, 2, and 3 are the receptor binding motif, or the virus's receptor binds. 6 and 7 are the interface of the receptor binding domain. 4 and 5 are the outer face of the receptor binding domain. And then you can subdivide those into smaller clusters with more precise biological behavior. If we look at those, and this is now negative state cryo-EM to get a broad sense of the array of structures and the commonalities and the differences by which these different therapeutic candidates will recognize spike, you can see things like this, that you can separate the ones against the receptor binding motif, for example, into flavors 1, 2, and 3 that approach the receptor binding motif from slightly different angles. Some are in the outer face, others are in the interface as the RBD lifts up. And we can do EM on the whole IgG and also the FAB fragment to understand how that bivalent IgG therapeutic might bridge different domains together, or when it achieves single occupancy versus multiple occupancy. The first thing we did was look at footprint analysis. We can map the footprint on the receptor binding domain made by the receptor in the dotted black line, and then the footprint made by each community of antibody in color. So RBD1 is a precise match for the ACE2 receptor binding site. And engineered molecules that are just receptor on an IgG-Fc are in that group as well. RBD2 shifted over here, RBD3 shifted over there. And then the ones against the inner and outer face may or may not overlap with that receptor binding motif or domain, or motif at all, or be shifted entirely off to the side. By mapping the footprints using the structural biology to get this global sense of the antibody activities now they're targeting, we can understand how they are or are not responsive to different variants that are emerging around the world. So if you take the vertical view of spike with one receptor binding domain lifted up, it looks like this. If you rotate the molecule around to the perspective of the target cell, there's the RBD sticking up. If we isolate to look just at the RBD, we can map the locations of major mutations that have emerged either by themselves in high frequencies in the patient population around the globe, or associated with different variants of concern that are emerging. Now, if you divide that receptor binding domain down by a line to separate what becomes the interface, where if you look at my hands in the picture, the receptor binding domains are at the top of spike. In order to interact with the receptor, they lift up. And so there's an interface, which is this part of the structure only exposed in the up. And then there's an outer face, which is exposed equally well if the RBD is down or the RBD is up. So this receptor binding domain has an interface and an outer face. And the communities of competition map to the different sides. So we see some that straddle, some that are clearly inner, and some that are clearly outer. Now the mapping that we have done to try to organize this global consortium meshes really well with mapping done by other techniques. So Pamela Bjorkman's lab and Ian Wilson's lab have done mapping and organization of different antibody candidates that can spike based on structural biology and based on the germline sequence from which the antibodies evolve. And it makes a lot of sense. Here we've done it by competition, physical biochemical competition, because we knew we needed to look for molecules that could together be cocktails. So for example, what Pamela Bjorkman in the Chris Barnes paper calls class 1 are RBDs 1, 2, and 3. They block the receptor very well. They bind sometimes bivalently, and they require an up RBD. And we can get that kind of information through the other molecule as well, and map these classes by competition across a very broad array from different contributors around the world to what has been in the literature from the other groups as well. When we look at that competition grid of all these candidate therapeutics, we think, how would we make a really effective cocktail that can mobilize to LMIC, to low and middle income countries? We can separate the antibodies into their different communities by the structures and the visualization of how they bind. And so this is very much a therapeutic effort from a structural biology lens, envisioned by and driven by structural biologists. And so if two candidate therapeutics compete in this grid of this COVID consortium, they're dark blue. If they don't compete, they're light blue. To get a little better idea how it works, we look at 50 example antibodies that go across all the communities and the subgroups. We can see things like this. So we've separated group 6 and 7 just physically by competition, that you could pair a 7 with the potent neutralization in that orange 2A group, but not a 6. Same reason why we've separated 4 and 5 into different clusters. They have different behaviors, and we can or can't pair them with different potently neutralizing ones from groups 2A and 2B. So for example, you could pair the potent neutralization of an antibody from the 2A with one from 4 or 5 or 7. But 4 and 5 can't be at the same time. Now, as you might expect, the structural biologist, the footprint that a therapeutic occupies in the surface protein of spike tells you to a large degree whether or not it's going to be knocked out by these mutations and variants emerging around the world. So the 2As, although they neutralize beautifully and you want them so badly, they get knocked out by the K417N or K417T mutations that have emerged in the world. But those in the cyan, blue, and purple groups, 5, 6, and 7, those are variant resistant, and those become very interesting for therapeutic candidates. We then mapped in my laboratory whether or not the candidates and the communities could be escaped by the different mutations, looking at them individually and looking at them by their clustered variants of which they emerge in the world. And in these kinds of plots, we're also looking at 2 different versions of the virus that spilled over and back to mink last November. And we can measure the fold change and how well these antibodies work in vitro neutralization based on all the different mutations. If I'm coloring the black brown, it's a total knockout. That mutation has lost activity in that antibody. If I'm coloring it turquoise, that antibody works better with that mutation. Gray, there's no effect. And what we see are that, for example, in the B1351 and P1, the South African and Brazilian isolates, they knock out a lot of the candidate therapeutics that have been proposed that are in the red and orange groups. Whereas those in 5, 6, and 7 remain effective. Those are also in the outer phase. We can also look at delta, and we find molecules that remain effective against the delta variant that's causing much concern right now. And so we can focus our efforts in COVID down to molecules that remain effective and durable against all the variants that have thus far emerged, so we can come up with an arsenal about what we might keep and mobilize in the future as we expect additional mutation emergence. Now, because we are structural biologists and we think about things in a geometric way, we can think about how that bivalent IgG recognizes spike versus the FAB that we had traditionally cut off to look at in isolation in order to achieve resolution. Spike itself is not purely rigid and absolutely upright. So this data from Hangzhou and Beijing shows us that spike on the surface of the virus waves back and forth, maybe 40 degrees either way. And so you might expect the spikes to move to each other in a way that might facilitate some IgG cross-linking. Well, that's kind of what we see. Those antibodies in community 1 universally tend to be able to occupy each of the 3 monomers in the trimer, with the IgG sometimes able to bridge. those in the potently neutralizing group 2 nearly always bind bivalently like goalposts with the two tied together FABs like this, like goalposts, and the FC upward. Those in group 5 are very interesting because they're slipped down to the side of the molecule. And what they tend to do is bridge different spikes together. And so I'm showing you two cartoons of the one, two spikes absolutely opposed. This is something that happens with soluble ectodomain on an EM grid. Maybe it'll happen to free viruses, but not spikes that are together on the surface of the virus. What could happen with the spikes together on the surface of the virus, or surface of infected cells side by side, is they're tilted slightly. Those group 5 antibodies tend to bridge those together. And often the ones in group 5 seem to punch above their weight by how we're measuring it in vitro. They operate better in the living thing than in the isolated cells in the tissue culture dish. So there could be a structural biology reason for that. So we can get a landscape of how these different therapeutics work, which ones are more or less likely to continue to be responsive to emerging mutation. We've been able to do partner studies when a company had a single antibody that was a good candidate, but they needed a complementary partner to make a cocktail to do that matchmaking. So that's what we've done in the first year. And the next year, we're going to do more complete variant analysis and keep track of the new variants that we're expecting to keep coming and emerge. And do more analysis tools on our database, from which anybody can come in and look at their own antibody or the aggregate molecules of the field as a whole. Now, a study of this scale is only possible with the contributions of many. So there are 13 different labs performing parallel analysis, each using a different expertise or different tool. There are 56 entities that have contributed their candidate therapeutic molecules that are being evaluated in human clinical trials or in preclinical molecules. Everybody in my lab took a couple of these molecules to try to do the array of structures that we needed. And we'd like to thank the Gates Foundation, GHR, and NIH for the complementary funding. And I'm happy to answer any questions.
Well, thank you very much, Erica, for this incredible talk and describing this amazing bringing together of the consortium and the power of that. I'm sure Erica would be happy to answer questions.
So Erica, Stephen here. I wondered if you'd comment on the antibodies that are in clinical use and whether any of those are going to be effective against Delta.
The initial major concern came from the ones that had been propelled forward for emergency therapeutic use are lost or lost in part if they're a cocktail against the B1351. The new data for Delta are still emerging, and there are still new candidates working their way up the pipeline. And so there are candidates that are absolutely effective against Delta, and they may just be 1 or 2 steps behind in the pipeline. And so the hope is that by the more comprehensive analysis here, we can keep a library of things, because there are some molecules that are knocked out by South Africa, but they've come back for Delta.
Sure, yeah.
I might have discounted them a couple of months ago, but now they look more important.
And we see the same thing with targeted anti-cancer therapies. You sequence a patient's tumor, you start the patient on a particular targeted therapy, they do well for a while, then resistance emerges, but suddenly that tumor is now sensitive to a drug that had previously been viewed as being off limits on the basis of the previous tumor sequence. So the notion there is sequence, treat, and repeat.
Exactly.
Sequence, treat, and repeat. And I think we may be seeing similar things with COVID. I noticed Jane Richardson has raised her hand. Jane, would you like to unmute?
I was interested in sort of the sociological process that you've done to this, and it's pretty clear how you blind things and get these results from the different labs. But I wondered what was in your system once you find, for instance, 2 things from different labs that might work as a cocktail together. How do you get those partners together in a good way?
Great question. So I am blinded, and that is to ensure fairness of the study, that what I propose for future cocktails is based purely on data. and to make sure, just to ensure to all the companies that whatever paper is written describes general things, or antibody 47 instead of particular molecule from a particular place. But our program managers are unblinded. And so what will typically happen is that a company writes me and they say, we have a molecule that needs a partner. And my program manager, who has access to the data, will say, look at the aggregate table and say, here are 5 that we think will pair well. And they can share the blinded data on the 5. And so tell the company, look at 28, look at 145, look at 36, here's what we recommend and why. And they'll pick who they want to speak with, and then the program manager will connect them, and they'll have an offline conversation about whether they can make the agreements work to put that cocktail together.
Neat.
Yeah, and for some of the, most of our contributions are from tiny biotechs, little startups, and for them it's a tremendous opportunity because they might have a fantastic molecule, but they don't have the resources to run a clinical trial, and they don't have a BSL-3, and they probably don't have the, all the different kinds of assays we have. So they're getting back this data and they're getting the opportunity. And when a big company wants to pick it up for them, they're quite happy.
Yeah, I think, and it's a great way to leverage potentially a very small investment. Ethan Cartagena is asking, you mentioned often that competition problems arise when antibodies are paired together. He would like to know why it's necessary to pair them in therapy.
Viruses have minimal proofreading and are constantly propagating errors. And if they have an error or mutation propagated that allows them to escape the pressure of one molecule and one footprint, they will do so. Nearly every antibody I've ever seen can be escaped. Eventually, it is the only thing there. By having 2 different footprints, you're requiring the virus to spit out 2 different mutations in the same place, unless there's something that gives an allosteric effect. And so it minimizes the likelihood of escape, and it maximizes the time of treatment in order to get a mistake, in order to spin off escape. You might get that patient cleared before the statistical likelihood of that multiple mutation event would happen. And then if you have a cocktail in your arsenal, it takes a long time to go from lab to clinic to approval to being in a doctor's hands to use. The virus will mutate in the world faster than we can move that pipeline along. If you have multiple cocktails, you will still get some benefit if 2 of the 3 or 1 of the 2 are responsive, whereas treating with a monotherapy that doesn't work is dangerous because it'll inspire additional mutation and it may provide no benefit at all to that individual patient.
Right. Greg Critchlow from RCSB Protein Data Bank asks, is clustering or reorganization of spike proteins observed upon exposure to your type 5 antibody? Could the angle changing affect an unproductive face of the spike for binding receptor?
That is a great question. We haven't looked at that, and maybe we should. That if we pressure treat with something that will better angle the movement, how does that affect viral activity? Now, these kinds of antibodies can have moderate to good neutralization, never typically awesome or the best, yet they can confer protection in the animal model at a much lower dose. So there's something good that's happening there. And it might be that we're not measuring it in the one single monodispersed cell type we have. Like
sure.
Maybe
yeah.
You can't pick it out in virial cells, but you can pick it out in whole animal, or maybe it has to do with the immune system, or maybe it has to do with cell-cell fusion, some other physical mechanism.
Thank you. Natalie, there's one more question in the chat. Should we do that and then move to the speaker panel?
Sure.
Okay, so TJ Ride asks, do you think a strategy like this could be used to develop improved flu vaccines?
Quite possibly. Anytime in science when there is a bigger black box to disentangle than any group and their tools can alone, we need some structure to enable that to happen. And even though we intellectually know that this is what ought to happen, you have to make it possible, right? You have to make sure the investigator can still publish and the student can still get their degree and people are going to get first author papers. And so you have to come up with systems and structures that protect all of those things that we need in order to enable that broader view to happen.